Great. Well, good morning and good afternoon, everybody, and welcome to the second in our series of Grants for Ag seminars, where we're delighted to invite our Grants for Ag awardees to tell us about their science and tell us about the work that they're doing. And I'm really delighted this morning here and this afternoon in Europe to welcome Dr. Ingrid Span to our program. We have gotten to know Ingrid over the last few months as part of our Grants for Ag program as a professor at the Heinrich Heine University in Düsseldorf, but that's all about to change, and I know she's going to tell you about that in her remarks now today. But she's going to spend some time talking to us about DNA zymes, the topic that she works on with her lab, and thinking about them in the context of a tool to fight viral diseases. So something that's of importance to agriculture and to Bayer as a company at large, and I know we're excited to hear all about that. Before I turn the floor over to Ingrid, just a quick note for those of you who haven't heard about it and aren't familiar, but this series is brought to you as part of our Grants for Ag program. And this is an opportunity we have within the crop science R&D organization within Bayer to provide essentially seed funding to innovators to test a proof of concept or develop their first idea out of the gate to think about an application that could be agriculturally relevant. So with a little bit of funding from our side and connection and mentoring from Bayer scientists, our hope is that we can accelerate the discovery of products and opportunities that could be really impactful as we think about technologies and advancements for agriculture, on a global scale. So we actually this week, so hot off the press, settled on our awardees for our 2022 program. So be on the lookout for the news of who's coming next. But at least for right now, we're excited to have Ingrid with us today, who's going to tell us all about her science and her exciting news going on in her lab. So Ingrid, I will stop sharing my screen, and if you would care to share yours, and the floor is yours.
Thank you. Just a minute. Okay. Well, hello everyone. Thank you for inviting me to the second seminar of the Grants for Ag series, and I'm happy to share some of the news that were happening in my lab. So the talk will be about fighting viral diseases in agriculturally relevant crops with DNA zymes. But first of all, I would like to share some personal news. So this research in the past 7 years, I've been working at the Heinrich Heine University in Düsseldorf. So this is the nice botanical garden that is just next to our building, and this is the campus. However, last week I was appointed as a full professor of bioinorganic chemistry in the Department of Chemistry and Pharmacy at Friedrich Alexander University in Erlangen. So this is the main building. So in the near future, my lab will be moving. However, as all the work so far has been done at Heinrich Heine University in Düsseldorf, I have decided to stick with the Heinrich Heine design one last time. So this is the last presentation in this design for myself. So I would like to start the presentation with a little bit of big picture and introducing the 7 sustainable development goals that were made by the United Nations. These goals aim to safeguard life on Earth and improve the world that we live in. And they involve, for example, no poverty, zero hunger, good health and well-being, quality education, gender equality, clean water and sanitation, affordable and clean energy, decent work and economic growth, industry, innovation and infrastructure, reduced inequalities, sustainable cities and communities, responsible consumption and production, climate action, life below water, life on land, peace, justice and strong institutions, and partnership to meet these goals. So when these development goals were published, I just started wondering how our research contributes to achieving these goals. So currently projects in my lab are focused on contributing to two main goals. One is good health and well-being. So we are interested in DNA enzymes. That will also be the topic of this talk, but in the context of therapy for neurodegenerative diseases and for treatment of HIV infections. Other projects are structure-based drug discovery for the treatment of cancer and the treatment of multi-resistant bacterial infection. So all these projects contribute to ensuring healthy lives and promote well-being for all at all ages. Another focus of our lab is the biological hydrogen production, as molecular hydrogen is a zero-emission energy carrier. This project fits into the goal to ensure sustainable and modern energy for all. Some of the projects that we work on also fit in the Sustainable Development Goals, aiming at conserving oceans, seas, and marine resources. In particular, our efforts to understand alkane oxidation in microorganisms that are able to degrade the components of crude oil. They could contribute to the field of bioremediation. One possible application that we envisioned is to help cleaning the oceans after major oil spills without disturbing the ecosystem significantly. Our research towards developing biological hydrogen producing devices also contributes indirectly to combat climate change and its impacts. And the Grants4Ag program of Bayer has inspired us to think about how we can use our knowledge and skills in the agricultural research and contribute to minimize food losses. So here we developed the project Fighting Viral Diseases in Agriculturally Relevant Crops with DNA enzymes. So as mentioned before, DNA enzymes was part of our research interest, but guided towards human health. And now we started thinking about applications that are in the agricultural sector. So here you can see an overview of the most important food crops. Depending on the source and parameters used for this classification, we find that sugarcane, wheat, rice, and maize are among the top 4. In some studies, sugarcane is on number 1. In most of them, maize is on number 1, again, depending on the source. Also in the top 10 are white potato, sugar beet, barley, and sweet potato. So as a chemist, I thought that barley and wheat looked very similar, so I decided to use a picture of a popular product of barley instead of actually the plant. So next, I would like to show you why the threat of plant viruses is increasing. Plant viruses cause considerable economic losses and are a threat for sustainable agriculture. The main factors driving virus emergence are agricultural systems that are based on cultures with low genetic diversity and a high plant density. These cultures are more vulnerable to pathogens and pests in general, and the high population and the increasing demand for food somehow contributes to larger agricultural systems. The second point is the world trade of plant material that moves viruses, hosts, and vectors to new regions and environments. The third factor is the climate change that affects the distribution area of hosts and vectors, and increasing dryness and heat somehow accelerates in some areas the occurrence of viruses. And the last part is the ability of viruses for rapid evolution and adaptation. So all these factors contribute that viral infections for plants have become a more dramatic situation. For our Grants4Ag project, we have decided to target the maize dwarf mosaic virus, short MDMV. This virus is part of the Potiviridae family and is a serious maize pathogen. It consists of a single-stranded RNA virus, which is of importance for our project as we work with a DNA enzyme that is capable of cleaving RNA strands. The vector for this virus or virus transmission are aphids. and the virus is epidemic worldwide, one of the most common viral diseases in grass-like plants, causing up to 70% of loss in corn yield globally. The virus has severe implication to the corn plant's development. So this affects the size as well as the stability and the metabolism of the plants. So especially the ear formation and development are slowed, and leads to significant grain yield loss, which again results in reduction of the plant size and weight, and also leads to these chlorotic spots on the leaves, which may eventually turn into yellow streaks, and at some point they have a mosaic pattern, and this is why it's called a dwarf mosaic virus. When plants are infected with the virus, there's no cure for the viral infection. Unlike bacteria or fungi that can be treated with antibacterial or antifungal agents, there's no treatment for viral infections. So disease management relies on getting plants resistant to viral infections using multiple strategies that must be developed specifically for each virus, each host, and each environment. An alternative option is to prevent the viruses from entering the plant, and we think that we can use our DNA enzymes that are able to cleave RNA in an efficient and highly selective manner to degrade the viral RNA before it enters the plant. So the DNA enzyme shown in blue can bind to the MDMV specific RNA, cleave it, and then the virus is not able of entering the plants anymore. The DNA enzyme that we focus on has been initially selected to cleave HIV, so the human immunodeficiency virus. And it's able to cleave more specifically the RNA of HIV. Therefore, we know that viral RNA is a good target for this specific DNA enzyme. Targeting the RNA before it enters the cell is also a particularly attractive strategy, as DNA enzymes are less active inside of cells. But I will show you some of the data on this particular topic in a minute, and also how we try to understand this phenomenon. So at this point, I would like to give you a quick introduction into DNA-mediated catalysis. So here you can see 3 of the 4 building blocks of life. So we have DNA, which is predominantly the molecule responsible for storage of genetic information. We have RNA, the carrier of genetic information, and proteins, the functional units inside the cells. So proteins are the most common functional units or biocatalysts in nature, and they are responsible for all the catalytic processes inside the cell. However, RNA can also form an unusual 3-dimensional architecture and perform catalysis. These so-called ribozymes are believed to be the first catalytically active biomolecules and play an important role in evolution. Most people know DNA in the double-stranded form, as mentioned before, the molecule that stores genetic information. These are particularly stable molecules, which is the basis of life, that DNA is not destroyed easily. However, about 2 decades ago, it was discovered that they can also adopt unusual 3-dimensional conformation, as shown here, and then perform catalytic activities or chemical transformations. Most of these reactions involve nucleic acid substrates, so DNA or RNA, and consists of cleavage or ligation reactions. However, the scope of DNA-mediated catalysis has broadened during the past years. So this is the DNA enzyme that my group focuses on, the 1023 DNA enzyme. What do we know about DNA enzymes or DNA catalysts? So they consist of single-stranded DNA that can adopt an unusual conformation resembling that of ribozymes. They have not been identified in nature yet, but they were obtained by in vitro selection methods. So the last seminar, Maria introduced the SELEX method, and I think her group also explained it in a dance. So this method was also used to select these catalytically active DNA zymes. Initially, they were conceived to function as gene silencing agents, but the reaction scope and applications have broadened significantly. The 1023 DNA zymes consists of a catalytic core. These are 15 nucleotides that do not form base pairing for the most part, and it has 2 binding arms. So these 2 substrate binding arms can be designed to bind virtually any RNA sequence. So here in red, you see the RNA substrate, and depending on the target RNA, the DNA zyme binding arms have also a complementary sequence. The cleavage happens between 2 nucleotides. At one of them is unpaired and one of them is paired. So in the case of the 1023 DNA zyme, the cleavage happens between an unpaired purine and a paired pyrimidine. And one important feature of this DNA zyme is that its catalytic function depends on divalent metal ions. So what applications have been shown for DNA zymes in general? So 2 major application areas of DNA zymes have been described so far. The first one is therapy. DNA zymes have been investigated in terms of the treatment of disorders that are related to elevated protein accumulation, viral infections, and prion disorders. So the research on prion disorders is particularly popular in our institute. So this is the main target that we have been working on so far. So prion disorders are neurodegenerative fatal brain disorders that can occur in humans and animals. The most prominent PRP-related disorders are Creutzfeldt-Jakob disease, mad cow disease, and scrapie in sheep. The 1023 DNA zyme, or variants of 1023 DNA zymes, have already been in preclinical model studies and a small selection in clinical trials that focus on the treatment of basal cell carcinoma and asthma. PRP disorders are characterized by the formation of large protein aggregates. By reducing the mRNA level of the mRNA specific for this protein, this leads to a reduction of the protein levels and subsequently a reduction of the aggregate formation. So by cleaving RNA, PRP-specific RNA, and reducing or lowering levels, not completely to 0, but to just a lower amount, this translates from the RNA into the protein area, and then the aggregation is highly dependent on a high protein concentration. So by lowering the concentration, the aggregate formation can be basically impeded. So another area that is a little bit newer but highly exciting, in my opinion, is biosensing. So there are DNA zymes that are capable of detecting metal ions with very high specificity. And another very exciting application is the sensing of microbial contamination. So this specific example, an RNA cleaving DNA zyme specific for E. coli was designed and coupled to a fluorescence signal. So basically, the setup is capable of detecting in real time using a fluorescence signal whether E. coli is present or not. And this construct was then immobilized on a transparent polymer that can be then used as a wrapper, for example, for meat. And this is an apple, if you don't recognize it. So basically, when E. coli contamination occur on this polymer, you will get a signal, a fluorescent signal that you can then detect. So you would know not to open things that are contaminated by bacteria or other microorganism. However, there's always a however. So despite the great potential and progress in the field of DNA science, there is a major challenge. And this is the activity of the DNA zyme in vivo. It has been shown that the DNA zymes are capable of effectively cleaving full-length in vitro transcripts, so full-length RNA. However, they fail to lower the RNA levels in cells. So I want to show you these results by a collaboration partner in our lab. So this is a work by Julian Victor, and he has tried to demonstrate the catalytic activity of DNA zymes inside cells for years. We have been working with a PRP mRNA-specific DNA zymes because the system was established at that time in the lab. And using urea page, he could demonstrate that the DNA zyme 839 that was designed to bind and cleave this specific mRNA is indeed capable of cleaving full-length PRP transcript into smaller cleavage products. So from this results, we identified a suitable DNA zymes, which is called 839. And then this DNA zyme was investigated in cells in WAC2 neuroblastoma cells. And we have here untreated cells. Then we have a positive control, which consists of small interfering RNA that is designed to specifically bind to the same region as the DNA zyme. And as we can see, the levels are lowered here. The protein levels are lowered. And this shows us that this specific area is accessible for the small interfering RNA. So the DNA zyme was not capable of reducing the protein levels, and also the negative control, which consists of a small interfering RNA that is not specific for this mRNA, has also not, as expected, showed a negative result. So this showed that the performance of the 1023 DNA zyme in vivo is very low or not detectable, whereas it's very active on the same RNA in vitro. So this has been one question that we have to try to work on in the last couple of years. And today I want to show you some results from a previous student, Hannah, who focused on the role of metal ions in DNA catalysis, and some recent work from my student, Nina, who investigates molecular crowding conditions and how they influence DNA catalysis. And then I want to just show you just briefly what we were able to do to design or to start the project if we can use 1023 DNA enzymes to target MDMV RNA. So the catalytic performance under conditions resembling the cytosols was our goal to investigate. So we used this 1023 DNA enzyme specific for PRP, and we know that this depends on the concentration of magnesium ions in the cell. And the next thing that we wanted to investigate is whether monovalent ions such as sodium or potassium also influence the catalytic activity. We investigate, or our method to investigate the activity of DNA zymes uses thread-based assay, so first the resonance energy transfer. So we have a donor molecule and a quencher molecule, and in the complex of RNA and DNA zyme, so the RNA is depicted in red and the DNA zyme in black, we have a quenching of the fluorescence, so we see no signal. If the catalytic reaction is performed by the DNA zymes, the RNA is cleaved and released, and when these two parts drift apart in solution, then the donor and quencher distance increases, and we observe a signal in fluorescence. So an increased fluorescence signal is a hint of activity or correlates with the amount of free RNA in the solution. So this is an example of the data, the normalized data from a FRET-based assay. So with we observe an increasing fluorescence, which means that the DNA enzyme is active under these conditions. And what we did now is we tried different concentration of sodium chloride. And some of you may think, why sodium and not potassium? Because the concentration of potassium is much higher in the cell than of sodium. However, we also repeated these experiments with potassium and saw the same trends. Since most biochemical assays are performed in sodium chloride, I'm still going to show the sodium chloride data. So we have concentrations of 0 millimolar sodium chloride, and we see a very fast increase, so a high activity. And if we increase the sodium chloride concentration, we see that the activity becomes lower and lower. So because this part at the beginning is not as steep, it means that the reaction is slower, the reaction rate is slower. We can also depict all the data that we have for all the different magnesium concentrations and sodium concentrations in a three-dimensional plot. And basically the result of our study is that higher ionic strength or higher concentration of sodium and potassium inhibit magnesium-mediated catalysis. So the DNA enzyme, under conditions that are close to physiological, is inhibited, or the activity is decreased at ionic strength that resemble the cell. So this is not good. Then we went a step further, and we also investigated the DNA enzyme in presence of manganese, because it's also known that manganese leads to activity, or the presence of manganese also is capable of restored DNA enzyme activity, and the activity is even higher in the presence of manganese. However, it's not as biologically relevant because the manganese concentration inside the cells is not very high. So in the same FRET-based assay setup, we investigated now manganese at different concentrations of or increasing sodium concentrations, and we observe that in this case, so the light curves are low sodium concentration and the dark curves are high concentration. That with manganese we observed exactly the opposite trend. So at low concentration we have a lower activity of the DNA enzyme, and it increases with sodium. So this was interesting. This is another representation that depicts how the rate constant K-ops increases with increasing sodium concentration. So this study showed us that the sodium concentration accelerates manganese-mediated catalysis. So our conclusion is that sodium levels have opposite effects on magnesium and on manganese catalysis. Unfortunately, magnesium is the one that is present in the cells at higher concentrations and not manganese. So another thing that we were interested in, we saw that divalent metal ions influence the activity of the DNA enzymes. But we were wondering how many magnesium do we need for one DNA enzyme RNA complex? So how many metal ions bind to one complex? For this study, we used RNA substrate that is stabilized. So it means that the 2OH group is exchanged by a fluoride group. So we are not able to detect any catalysis. By inhibiting the catalytic turnover, we can investigate the properties of the DNA enzyme RNA complex in more detail without having to care about activity or like cleavage reactions. So we characterize the metal ion binding using ITC, isothermal titration calorimetry, where we see the development of heat in a positive or negative direction during the reaction. And using this method, we could first of all say that the binding of magnesium to the DNA-zyme complex is weaker at higher sodium chloride concentration. So this again points to an unfavorable conditions inside the cell for metal binding. And we also know that we need multiple binding sites. So we need more than one magnesium ion for catalytic turnover. So this was the first part where we looked basically at the metal ion concentrations. The second approach that we have to understand why the DNA-zyme is not as active inside the cell as it is in vitro is to look at the activity or the catalysis under molecular crowding condition. So the catalytic function of all biomolecules is different in the crowded environment of a cell that is represented here. So inside the cell, it's not the same conditions and we have inside an Eppendorf tube, for example. So this is much more crowded than the dilute conditions of in vitro assays. The presence of molecules such amino acids, polypeptides, alcohol, and sugars in the cell introduces forces that modify DNA-zyme structure, folding, as well as catalysis. And it's been reported that we can use synthetic molecules to mimic these conditions and hopefully get a better understanding of why in vitro and in vivo conditions or results are so differently. The first thing that Nina looked at is the activity and stability of DNA-zymes and the RNA target by urea page. So we use polyethylene glycol, so PEGs at different concentration. This is just an example. And then we also use osmolytes to investigate molecular crowding condition. So at this size, you can observe the uncleaved target, and this is the 5' fragment of the cleaved target. And the first lane is always just the target, so the RNA. And we are worried about RNA stability because RNA in general is not as stable as DNA-zyme. So these are just transient molecules. And you have to be very careful when handling RNA. So our first concern was, does the RNA survive our assay or treatment? And as we can see, in all samples, the RNA is stable in the presence of these molecular crowding agents. And the second lane represents the target and the DNA-zyme. And here we wanted to see if the DNA-zyme is capable of cleaving the target in the presence of this crowder. And for all samples, we see that the DNA-zyme is also able to perform catalysis. That is good. So the next step is then to investigate the effect of the molecular crowding environments on the kinetics of DNA-zyme catalysis. So we used, again, a FRET-based measurement setup. And here you can see the mass concentration, so the amount of crowder in our reaction and the reaction rate that has been normalized. And we see that it's not as straightforward, but most of the molecules seem to decrease the catalytic activity of the DNA-zyme. So increasing molecular crowding, which resembles the state or the conditions inside the cell, does not have a positive impact on DNA-zyme catalysis. So this is just a very small subset of the data, but we looked at different molecular weight crowders, so polyethylene glycol. We looked at very small ones up to 20 kilodalton. And then we also looked at different mass concentration in this dimension. And then you can see the areas that are colored in red are high catalytic activity, so a high reaction rate, K-ops, and the area of blue are low catalytic activity, so low reaction rate, K-ops. So what we can see is that it's not as straightforward, or the trend is not as clear. However, in general, we can say that higher molecular weight and more crowded condition, so this corner, decreases the activity. So the size of the crowder as well as the mass concentration, or increasing size and increasing mass concentration, have a negative impact on the catalytic activity. In addition, we also investigated osmolytes. Osmolytes are low molecular weight organic compounds that influence the properties of biological fluids. And here we observed a little bit a clearer trend that increasing mass concentration leads to lower activity. Therefore, this area is clearly blue, and the lower mass concentration leads to high activity. So in conclusion, our results show that crowded environments affect DNA enzyme activity, and that increasing concentrations lead to a lower catalytic activity. And we're trying to find out whether the structure is altered or the physical properties of the solution, or what the reason is. And we don't have a clear picture yet. So now I would like to show you the first things that we've done with the MDMV specific DNA enzymes. So in theory, everything that we found out indicates that DNA enzymes are not as active in the cells as we would like them for therapeutic application. However, in the cases of virus treatment, the goal is to prevent the virus from entering the cell. So we're not worried too much about this low catalytic activity inside the cell. And what we've done so far is that we have designed DNA enzymes specific for MDMV, and therefore we have identified regions in the MDMV RNA that are suitable. So the criteria that we need is the recognition sequence, a purin and a pyrimidine. And we also need to consider that RNA has secondary structural elements, and they are not the same as in proteins, of course, but it can also form hairpins and loops, and therefore we need to predict using bioinformatics if the region of the RNA around this pair is accessible for DNA enzymes. Another thing that has to be considered when we design the length of the binding arms of the DNA enzyme is the stability of the DNA enzyme RNA complex. It has to be stable enough for the catalysis, but also the dissociation constant of the complex after the reaction, because an efficient catalyst has also to release the product really fast. So this is also a requirement. And we have finished the design and have identified sequences and designed potential DNA enzymes, and we're ready to test them now according to our previous thread-based assays. So taken together, I hope that I could show you that DNA enzymes are super exciting and have a great potential. So they consist of single-stranded DNA molecules that catalyze chemical reactions. The RNA-cleaving DNA enzymes have a great potential for biomedical and biosensing applications, and hopefully we can use them in the agricultural research area a little bit more in the future. The activity of the 1023 DNA enzyme has only been demonstrated in vitro, and the high levels of sodium and potassium and the low levels of magnesium inside the cell decrease the activity, and also the molecular crowding environment in cells decrease the activity of the DNA enzymes. However, for applications for the treatment of plant viruses, this is not too dramatic, because the treatment or the degradation of the RNA should hopefully happen before the virus enters the cell or the plant. And we have finished the design of the DNA enzymes specific for the MDMV. And the next step is to perform activity assays in vitro using small fragments as well as full-length RNA. And we are looking for a partner to help us to determine the activity of infected plants. So now I would like to acknowledge my students Nina and my former student Hannah, who have done all the work that I've showed you. We also have great collaboration partners, Gerhard Steger, Detlef Riesner, Manuel Etzkorn, Julian Victor, Jan Borggräfe, Aldino Viegas. They all used to be here at Heinrich Heine University Düsseldorf, but also Forschungszentrum Jülich, Ralf Biel, and Olaf Schiemann have helped us a lot in an area of structural determination, which I haven't presented today, but we also try to solve a structure of the DNA enzyme to better understand the mechanistic of DNA-mediated catalysis. Of course, I would like to thank Bayer for the Grants4Ag funding. We also have funding from Fonds der chemischen Industrie, Deutsche Forschungsgesellschaft, Forschungszentrum Jülich, and the German Ministry of Research and Science. This is my group at Heinrich Heine University now. So as I mentioned, we have a beautiful botanical garden just next door, which we like to use for our group pictures. And for everyone who missed the last Grants4Ag seminar by Maria on combining nanotechnology and molecular recognition for agriculture and food, here's the QR code to her talk on YouTube. And if you want to know more about the old Grants4Ag awardees, or maybe this links already to the new ones, then you can use this QR code. And last but not least, I would like to thank everyone for their attention, and I'm happy to answer questions.
Thank you, Ingrid. I wouldn't call them old Grants4Ag awardees. Maybe senior.
Yes.
Thank you. So yes, questions for Ingrid. I see Tobias has dropped on in the chat, but anyone's welcome to come on camera, and it's a small enough group, so please turn your camera on, unmute, and ask your question.
Hey Phil, hey Ingrid, thanks for the presentation. I'm on my way to picking up my son, so I won't come on camera. However, the question I have is about stability of the DNA enzymes. Are there any limitations that you can foresee when using them in crop applications?
Thank you. That's a great question. Actually, that's something I usually like to show, but I wasn't sure if I have enough time. But now I can maybe take a minute or two and talk about the stability. So in general, DNA has a lot of advantages over RNA and over proteins. So as DNA was or is the carrier of genetic information, it has to be incredibly stable. And this is just due to a small chemical modification, which is just the lack of this 2OH group. However, this makes the molecule way less labile against chemical degradation or hydrolysis. So in general, DNA is super stable, and you can really you don't have to have any precautions when working with DNA. It's more hard to get rid of it. So I think in terms of stability, DNA has superior characteristic versus proteins or RNA. So when working with RNA, you have always to be very careful about RNases and degradation of RNA, and their stability is incredibly hard. So I think this is one of the major advantages of using DNA enzymes, for example, over ribozymes, is that their chemical stability and also against degradation by enzymes is super high. And there's no such thing as like DNases. I mean, there's such a thing, but these are not very common because it would like kill all the organisms. Of course, we have it in the lab, but so I think that if you're thinking in terms of applications in the field, then RNA I think is super labile and will not be a problem. However, DNA is very stable, and we are very confident that this can be also applied in the field or on plants directly. And they're also much more stable than proteins, and RNA interference techniques have also a lot of other downsides. They need like helper proteins, for example, which we don't need with the DNA enzymes. DNA enzymes are fully self-sufficient biocatalysts, so they don't need any helper proteins.
Awesome, thank you. Other questions for Ingrid? Folks, please turn your cameras on, unmute, and ask away. No? Anyone? I have one, but I'd love for someone else to jump in. Or maybe Ingrid, I'll ask a question. Oh, Peter, please do go ahead.
Yeah, thanks. Just put my camera back on. Yeah, I'm sorry if I missed something there, but molecules like DNA are not that stable, are they, if you get them out in sunlight? What might be the methods of using them in real field conditions?
So I think, I mean, highly intense UV radiation is of course used to degrade DNA molecules. But I think in general, the stability is still pretty decent. We haven't tried this, and I don't really know of techniques that have been used or like studies that have been used to really determine this in daylight. But I think in general, the DNA molecules should be fairly stable if it's not under high radiation conditions. I could still imagine, like I talked to Maria, for example, if we can somehow combine our expertise and skills and have something that sensors, so they have like these particles where you can release substances up in detection of like changes in the environment or something like that. So I could also imagine that this could particularly be a method that we have to, like, that we use somehow of a protection or something. But I think that these in biosensing, they use really these polymer films and just in like normal daylight, and they have not observed any degradation so far, like on a large scale, that it's not active anymore. So I don't know if this could be somehow, if this is really a problem.
Yeah, I'm just wondering how this technique might actually be applied in the field. Do you have any ideas or any experimental data to indicate that?
I mean, we just really have experience in the basic science, and we have never really thought this is really a very new idea to go into the agricultural sector at all. And this is one thing that I mentioned in the summary and outlook sections, that we're also looking for collaboration partners that have a little bit more experience on like testing this in the field. Because yeah, I think this is still very new. I think DNA catalysis is still a very new field compared to others. But we really don't have a good idea on how this would behave.
Yeah, I suppose another question related to practical application is that in agriculture, of course, you need to apply over a huge area. You know, just imagine how many maize plants there are in the world. You said at the beginning that the disease is quite widespread. So how does the production of this kind of DNA scale up?
I mean, I think DNA production is super cheap in comparison to like RNA sequence. So I think it's for our constructs and our length, it's a factor of 10 for the RNA almost than for the DNA. So DNA enzymes really just a few euros. So I think it's not like from a financial cost of view or a point of view, it's the costs for DNA are way lower. This is also because of the stability than for RNA. So I actually can think of like also using this as a detection method. And I think the application is only required if you really detect the virus. So I wouldn't imagine to use this as a standard procedure. But if you detect infection, this usually spreads very widely. And I think in this case, maybe it could be useful to combine it with like a diagnostic, which is also, I think, an area that could benefit from more research, the like simple diagnostics of viral infection. I know that there is a kit actually for this type of virus, but I think for other that are not as common, DNA science could be maybe also designed just for detection purposes. So that's just a very recent idea that we thought about.
Yeah, thanks. It's really interesting that we've got an approach here that works against viruses, which, you know, really tough problem in agriculture, as you said at the beginning. And so far, we've only been able to fight the vectors. So yeah, it's great work, very interesting. Thanks. And I live in Düsseldorf, so I know the Botanic Gardens. Yes, I'm sure you'll be sorry to leave.
I am. Yeah, so I think we were also surprised when we looked into this area that it is at the beginning we just had this idea and we just thought, well, if we use human RNA, viral RNA, no, if we target viruses that attack humans like HIV, why not doing that for plant viruses? But we weren't even sure if this is a problem. And then when we saw, but it's really, I think it's not a big research field, surprisingly, even though I think it's such a dramatic problem and it increases steadily. Yeah.
Thank you.
Well, Peter, you did a great job there, because you basically asked the exact same question I was going to, in terms of next steps and translation and scale. But I know, Ingrid, something we can certainly talk about with the Bayer team in terms of what we might be able to do when the Grants4Ag program is over. Yeah. Maybe last call for questions from the group. Okay. Well, in which case, just remains for me to thank you, Ingrid. I really appreciate you taking the time and your engagement in our program and the great science you're doing. So maybe a virtual round of applause for you. So thank you so much. And thank you. All the best of luck with moving your lab. I know that's not an easy thing to do, especially in pandemic times. So best of luck with that. And we look forward to staying in touch and continuing to work together. But,
me too.
On behalf
of
the group, thank you.
Thank you, Phil, for putting this together.
Very welcome. Very welcome. Great. And everybody else, have a good day, and we'll talk again soon. Bye-bye.
Bye.